Okay, thank you, Luis. Hello, I'm Randy Hubbell. I'm the President and CEO for Carmell Therapeutics. We are a clinical stage biotech company with a unique biologic that is designed and discovered to assist in healing, both hard and soft tissue, both bone and soft tissue. So we have a significant opportunity as we're moving our first product candidate into phase 3 clinical development, and we'll advance our second product candidate into preclinical and phase 2 as well. So a little bit about myself. I spent 13 years at J&J, last 5.5 at J&J Biosurgery, where we were able to get 2 biologic BLAs approved. With me is Dr. Hart. Dr. Hart is our Chief Medical Officer. Dr. Hart retired from J&J as Chief Medical Officer for J&J Global Surgery, and he spent a significant amount of time with the Biosurgery Group and was the executive sponsor for the 2 BLAs that we did get approved there, a fibrin sealant and a fibrin pad. Also joining us is Donna Godward. Donna is our Chief Quality Officer. Donna spent 6 years as a Chief Quality Officer for J&J's entire medical device and diagnostic business before retiring now as our Chief Quality Officer. Dr. Stephanie Kladakis is our VP of R&D. Steph is a PhD in tissue engineering. She's got significant experience of taking technologies from concept to commercialization. And then Dr. Janet Vargo also retired from J&J, where she was a senior leader developing a significant number of clinical trials, and she's our VP of Clinical Sciences. So what do? We're in the business of reducing the burden of healing. We accelerate the time to heal, we reduce complications, specifically infections, and we have human phase 2 data that has demonstrated that. Our new biologic is off the shelf, ready to use, no mix, no prep, and our first applications will be used in surgery, but it's a platform technology. It can be used in many settings, and eventually we'll have an injectable and other forms of it as well. So here's some ideas on where we can use it. Where you see in highlight is the programs that we currently have in development, and then the others are in research and or potentials based off of a substantial body of evidence that exists today regarding the use of platelet-rich plasma to accelerate the healing in these areas, specifically to deliver growth factors right to the injured site. So we'll start with long bone fracture, and I'll give you some data on our trial from a tibia study, a tibia fracture study, and the bone product they're called bone healing accelerant. We're also studying it for spine fusion as a way to enhance the fusion of the spine. And then our soft tissue product, we'll start with skin grafts, but it has many applications as well, including things such as hair regrowth or reduction of scars, some dermatological applications. So here's what makes our technology so unique. This is what's never been done before. Nobody has ever taken whole platelet-rich plasma, allogeneic plasma, from donors and processed it like a medical technology. So we're able to do the viral inactivation steps without killing the regenerative factors. Whole platelet-rich plasma has hundreds, if not thousands, of proteins in it that are associated with healing. The ones that are contained in the platelets and our growth factors are ones that we most hear about and have the greatest amount of research done, such as VEGF, PDGF, TGF-beta. All of that's contained in our technology, plus more, and we don't kill it when we do the viral inactivation steps. Also of significance is form, presence, and bioactivity. So through the process that we have patented, we're able to plasticize our material. So we're able to create a powder form after lyophilization. Essentially think of it as a resin. And that resin then can be made into different shapes. Our first product is a bone healing accelerant that is in a form of a putty. Our second product is a tissue healing accelerant that is in the form of a paste. We also have successfully molded a hard screw, as well as a patch and a sheet and a ribbon. All of them biodegrade, and all of them release growth factors as they biodegrade. And here's what's significant. Never has there been an opportunity to deliver platelet-rich plasma in a controlled manner so that it biodegrades and releases growth factors on a continual basis. And that's done because of the unique cross-linking with genopin. Genopin is a derivative of the gardenia plant. It is currently used in food dye, and that cross-linking protects the proteins and forces the enzymes of the body to take longer to work through the genopin to get to the platelet and have the platelet burst and release those growth factors. Other forms of trying to deliver platelet-rich plasma to injured sites results in the majority of the platelets being absorbed within the first couple of hours. And then lastly, bioactivity. This is an off-the-shelf, ready-to-use product that has lot-to-lot consistency. We manufacture it at our facility. Our manufacturing is complete, and we've actually manufactured 3 full-scale lots that will be presented to the FDA. We measure 12 different tests to determine consistency. 3 of those are related to the bioactivity and the presence of growth factors. Nobody's ever been able to do that before. So we have a substantial market opportunity, even in the 2 product candidates we're currently developing. So wherever this trauma fixation device is placed, particularly in patients that are compromised, such as diabetic, smokers, obese, and elderly patients, there is often a need to add an adjunctive technology to that hardware, and that's where we come in. There's over 1 million surgeries performed each year for the placement of hardware for trauma fixation. We're also looking at the spine market, which has a market size of over 700 million, and then rotator cuff repair. That's with the bone healing accelerant. The tissue healing accelerant will start with split and full thickness grafts in burn patients and other chronic wounds. We eventually would like to get to chronic wounds such as pressure ulcers, diabetic foot ulcers. We have some journey to go before we get there, but that's an aspiration for us. We think our technology would benefit those patients as well. So here's our phase 2 data. We conducted a clinical trial in South Africa with 30 patients, 20 treatment and 10 control. The South Africa's location was quite appropriate for trauma patients, as there's a lot of trauma, and one of the world's renowned trauma orthopedic doctors, Dr. Brian Bernstein. It was open tibia fractures. Both the treatment and controlled received standard of care. After the hardware was placed to fix the bone, then it was randomized. And the treatment group got received the product, which was taken off the shelf and placed into the cracks and crevices of the bone during the open procedure. Then it was closed, and those patients were followed for 3, 6, and 12 months. The highlights of the results was that we saw twice as many bones healed at 6 months. We saw a statistically significant reduction in infection rates. These patients are prone to infection because of the disruption of the soft tissue. So as the bone comes through the skin, by definition, these open tibia fractures have compromised healing environments because of that exposure. We also saw 4 times more patients had their wounds healed at 30 days in the treatment group versus a control. If you think about your shin bone, the soft tissue lies right on top of the bone, so it's also being exposed to the regenerative factors. Here's the results. If you look at the control, this is at 6 months. This injury is typically 9 to 12 months to heal, so we looked at an accelerated time point. At 6 months on the worst patients, which are called 3A and 3B on the Castillo-Anderson scale. The first line there you see, once again, both of these received standard of care, and the treatment group received standard of care plus the bone healing accelerant, which is called PBM, plasma-based bioactive material. And when you measured it by X-ray, you have 4 cortices when you have an AP and a lateral view, anterior, posterior, and lateral view. As you look at those 4, they're looking for bridging of the fracture line, and healed is considered 75% or greater bridging. In the control group, only 1.3 cortices met that definition. In the treatment group, 2.64. That did hit statistical significance in a very small patient population. When we did a post hoc analysis using an upgraded radiology scale called modified RUS, Radiology Union Scale for Tibia Fractures, and we married that with the clinical endpoints of weight bearing and lack of tenderness to touch to create a composite endpoint. All three needed to be true in order to be a responder. None of the control patients met that definition, and 36% of the treatment group met that definition in the most severe patients. That was what was presented to the FDA in a pre-IND meeting, and that is what they have agreed to is our phase 3 design. So there's a direct link between our phase 2 study and our phase 3 design. We did see a dramatic reduction in infection rate. Some of these patients in this study had external fixators, which are pen tracks. Pen tracks easily get infected, so severity was high. Frequency was high, although severity was low, but it's still striking that we saw such a dramatic reduction in the rates of infection for the treatment group versus control, which is also supported by a published animal study that we have done, a preclinical study. So the regulatory update, the FDA meeting was on January 10th, 2017. We asked multiple questions. Are we ready to move to phase 3? Do you agree with our phase 3 design? Do you agree our primary endpoint is both clinically meaningful and important? And the FDA did agree to all of those. So we have since then been working on CMC, which is our manufacturing. And I mentioned already, we're extremely proud that we were able to create all of those manufacturing instructions and validate them, all the release tests and validate them. And we have run R&D lots followed by 3 consecutive full-scale batches, which have all passed the initial test. So we're very excited about that. I tell you all that because that is necessary for the IND submission, which will occur in January of 2019. We'll conduct 2 phase 3 studies per the regs of a BLA. This is a bigger lift than a medical device approval, but at the end, we end up with something much more substantial because we'll have 2 clinical trials demonstrating reproducibility at the time of launch. So here's our corporate timeline. The first product, first indication, we'll have first patient in Q2 of 2019, last patient in by the end of 2020. We will follow every patient for 12 months, although the primary endpoint is a 6-month endpoint, at which point we'll have data reveal. It is an adaptive design, so we will get interim analysis along the way. We will also progress our second indication with the same product, so same SKUs that will be sitting on the hospital shelf. We'll be conducting preclinical using the Bowden model, which is a standardized preclinical model for spine fusion. It's a P-lift in rabbits. And then the tissue healing accelerant, we are conducting preclinical work on burns, and then we will progress to phase 1/2A. We have multiple levels of protection. We have 16 issue patents. The main patents go out to 2032. Also in the US, when you're granted a BLA for being first in a category, you're granted 12 years of marketing exclusivity. And we also have what I call practical exclusivity. This is not a 505(b)(2). This is not data somebody can borrow. So anybody else following us would have to do discovery, preclinical work, phase 2, phase 3. So in summary, we are on the path to a major financing round next year, which will be private or public. We're preparing for both very diligently. We currently have a convertible debt round open today. It's a $6 million round, with half of it in the bank. The reason we went for the convertible round is because it gives us the opportunity to have flexibility and to pivot and have optionality. And if the opportunity continues to progress for a public offering, we could cancel the convertible round or end it and move to a pre-IPO equity round. Or we could go right from our convertible round to another private round, or right from our convertible round into an IPO. So all of that is available to us. The convertible round is a $6 million round, 8% interest. All of it gets converted into equity on the next equity round at a maximum of a $20 million pre-money, or 20% discount to the next pre-money equity round, whichever is lower. Thank you.